To explain nanotechnology effectively, you have to bridge the gap between material science, what it's made of, and biological interaction, how it behaves in a body. The building blocks, carbon nanotubes, CNTs. You must understand that nanotech can be carbon hollow tubes. These are known as carbon nanotubes. Structure. Imagine a sheet of graphene, a single layer of carbon atoms, rolled into a perfect cylinder. They are incredibly light, but 100 times stronger than steel. The organic illusion. Because they are made of carbon, the same base element as human life, the body doesn't always recognize them as foreign in the same way it would a piece of plastic or metal. This is likely why people mistake the resulting structures for organic mutations or bugs. Electrical conductivity. These tubes can carry electrical signals, which is why they are used in smart materials and sensors. The color mystery. Quantum dots and sensors. The red, blue, and black colors seen in images of exposure often relate to the optical properties of nanomaterials. Quantum dots. These are semiconductor particles only a few nanometers wide. Depending on their size, they emit very specific, vibrant colors, like bright red or blue when exposed to light. Refraction. When fibers or tubes are engineered at the nanoscale, they can manipulate light. What looks like a colorful bug might actually be a structural polymer, reflecting light in a non-natural way. The Red Era, 1990s to 2026. During the initial trials of atmospheric spraying, larger quantum dots were often used. Because quantum dots emit light based on their size, these larger particles, approx. 5 to 6 nm, emit a vibrant red. These were primary tools for environmental mapping and sensor calibration. Understanding the coatings. The reason these materials are so hard to identify is due to functionalized coatings, masking. Nanotech fibers are often coated in synthetic polymers or lipids, fats. This mask allows the tech to mimic organic matter, making it nearly invisible to the body's immune system. Refraction. When these fibers are engineered, they manipulate light through refraction. This means what looks like a colorful bug under a microscope is often just a structural polymer bending light in a way that doesn't occur in nature. Interaction. These coatings allow the fibers to stick to human biology, weaving into tissue, to create the synthetic composites or entities that are being documented in my research. By aligning the atmospheric spraying timeline with the quantum dot color spectrum, we see a clear evolution: red for initial mapping, blue for biological tracking, and black for advanced coated masking. The Blue Era, 1990s to 2010. As the tech became more sophisticated, smaller particles, approx 3 nanometers, were introduced. These smaller dots emit blue light. This era matches the 2001 Morgellons reports, where blue fibers became a hallmark of the condition. The Black Era, 2011 present. Modern nanotech often appears black because of advanced carbon-based coatings and light-absorbing composites. These are designed for multifunctional use, acting as a shield for the internal tech while absorbing light to remain stealth. Nanomaterials versus biology, the interaction. When these materials enter a biological system, they don't just sit there. They are designed to self-assemble. Scaffolding. In the medical field, nanofibers are used to grow scaffolds for new organs. However, if unguided or environmental, these fibers can weave themselves into the tissue, creating the leech-like or fuzz entities you've filmed. These aren't necessarily living organisms in the traditional sense, but biosynthetic composites. Masking. Nanotech can be stealth. Particles can be coated in lipids, fats, or proteins, so the immune system ignores them, allowing them to accumulate in the body over years. Fibers are hollow and act like a vacuum, and can suck in pathogens they come in contact with. The scale. Can you see them? The invisible scale. A nanometer is 1/1000th of a meter. A human hair is about 80,000 to 100,000 nanometers wide. Visualization. You cannot see an individual nanobot or nanotube with a standard 60x microscope. What you are likely seeing are macro aggregates, thousands or millions of these fibers and particles clumping together, or the biological mutations, extra collagen, keratin, or fibers, the body produces in response to the irritation. Modern milestones, smallest tech. The MIT camera. Researchers have developed cameras the size of a grain of salt, which houses 3 computers that monitor this stuff in our environments. Nano-engines. Scientists have successfully built molecular motors and nanotrucks, single-molecule cars, that can move across a surface when triggered by light or chemical energy. CPU chips. We are currently at the 2 nanometers and 3 nanometers node in commercial chip manufacturing. This means the features on a computer chip are now smaller than the width of a strand of DNA, which is about 2.5 nanometers. The connection between the 1990s atmospheric shifts and the 2001 Morgellons reports aligns with the incubation era of nanotechnology. In science, there is often a 10 to 20-year gap between laboratory breakthroughs and wide-scale environmental presence. If large-scale exposure began in the 90s, the physical breakouts, or mutations, appearing in the early 2000s, would be the logical biological result of a body finally reaching a saturation point. While the labs focus on the good of medical nano, we are left to document the environmental reality they ignore. From carbon nanotubes to the saturation points of the early 2000s, the evidence is mounting. We don't just film the anomalies, we track the design. This is CyberBeats